If mitochondria are so central to cellular function, they may also be central to aging, which is what we will discuss in this module. As we discussed mitochondrial function and dysfunction in the previous module, we only sort of explored the consequences in a more or less disease context, but not specifically aging. The failure of the free radical theory of aging necessitated a new aging hypothesis that involves the mitochondria. One such attempt to link mitochondria, damage, and aging is the mitochondrial lysosomal axis theory of aging. As discussed earlier, damaged mitochondria get continuously degraded by the cellular garbage removal network, which includes the lysosomes. Lysosomes break down damaged mitochondria, which includes oxidation of their membrane fatty acids. One of the end products of this degradation process is lipofuscin, sometimes called a molecular aging pigment due to its brown coloration. The more damaged mitochondria are degraded, the more lipofuscin accumulates. This in turn leads to the eventual clogging of the lysosomal network, the inefficient removal of damaged mitochondria, and their persistence in the aging cell, which in turn may lead to the detrimental outcomes as we've discussed earlier. This hypothesis may explain what happens to damaged mitochondria and how that may contribute to aging. But it doesn't tackle the source of the damage, nor does it investigate whether mitochondria may be responsible for aging in the first place. We thus need to dig deeper. Clearly, as discussed multiple times before, normal mitochondrial metabolism releases ROS. As the source of ROS production, mitochondria will also be the first target of this damage. But even without ROS, mitochondria are very susceptible to spontaneous damage accumulation. This can clearly be observed when mitochondria are stained for the presence of cytochrome C oxidase, an enzyme of the respiratory chain, often abbreviated RC. Other people call it the electron transport chain, or ETC. Mutated mitochondria that no longer express this enzyme will appear blue when stained with a particular staining method, instead of the regular brown. The source of this damage must be found within the mitochondrial DNA. Mitochondrial DNA exists as a single circular chromosome within the mitochondrial matrix, not unlike bacterial DNA. It contains all the genes for the respiratory chain, some rRNA and tRNA genes, while other mitochondrial genes have been outsourced to the cell nucleus. Mitochondrial DNA replication is carried out by a specialized DNA polymerase enzyme, the so-called DNA polymerase gamma, which only copies mitochondrial DNA and never nuclear DNA. In contrast to nuclear DNA replication, mitochondrial DNA is copied one strand at a time, according to the strand displacement model, and not semi-conserved as nuclear DNA gets copied. As part of this specialized process, the two new DNA molecules end up intertwined at the end of the replication, and therefore need to be separated. The entire process sometimes leads to DNA polymerase slippage, that is, the skipping of portions of DNA that need to be copied. Sometimes it can lead to incorrect breaking and joining of DNA pieces. Both result in the subsequent loss of mitochondrial DNA pieces as small extrachromosomal circles or plasmid-like DNA pieces. Thus, over time, mitochondrial DNA experiences deletions and gene loss, and the affected mitochondria will no longer express all electron transport chain genes, have impaired function, including higher ROS production, and all that entails. Another source of mitochondrial DNA damage is simply the consequence of the watery environment inside the cell. Spontaneous deamination of amino or NH2 groups on the DNA nucleobases converts an amino group into a carbonyl group. A practical consequence of this event is that it converts, for example, a cytosine base into a uracil base. Uracil normally is only found in RNA. These different bases, moreover, have very different Watson-Crick base pairing capabilities, which means that during replication, they base pair with a wrong base, and the newly replicated DNA will now have acquired a mutation. Interestingly, mitochondrial DNA damage is almost entirely from endogenous sources, that is, from within, while extracellular or extraorganismal mutagens have very, very little effect on mitochondrial mutation rates. The consequences of mitochondrial DNA deletions can be far-reaching. Loss of mitochondrial efficiency especially affects tissue types with high metabolic rates, such as the brain or muscle cells, which may be why these organs seem to be particularly affected by aging or aging-related diseases. Another consideration is that mitochondria are inherited via oocytes, and thus any mitochondria with damage or DNA mutations will be passed on to the next generation. Sperm cells do not pass on their mitochondria, and therefore an important balancing mechanism falls away. This in turn may negatively affect aging and disease phenotypes in that generation, quite possibly even contributing to familial longevity phenotypes. At what point do these damaged phenotypes become apparent? Interestingly, due to the redundancy in the mitochondrial pool, even a fairly high mutagenic load may often be complemented by healthy mitochondria or intact mitochondrial genes, which leads to normal phenotypes. But once the number of mutated mitochondria passes a threshold that is around 60 to 90% of all mitochondria, disease phenotypes become apparent. At this point, an almost normal cell will rapidly decline into a damaged, malfunctioning cell. One way to understand this threshold effect is to view the mitochondrial genes just like nuclear genes through the lens of genetics. In that sense, a recessive mitochondrial DNA mutation may simply reduce the number of properly functioning molecules, which would merely decrease efficiency. In contrast, a dominant mutation may completely abolish proper function of a protein or a protein complex, which could possibly stop the electron transport chain altogether. In extreme cases, such dominant mutations may even lead to lethality. Such lethal mutations may thus not only eliminate the affected mitochondria, but even the cells that carry them, therefore possibly contributing significantly to aging phenotypes. Mutations are by their very nature stochastic and appear at random spots in a genome. Thus, with hundreds of individual mitochondria within a single cell, each mitochondria may obtain a different mutation, which could be complemented by wild-type genes in other mitochondria. In reality, however, if one investigates mitochondrial mutations, it becomes apparent that mutations carried by mitochondria are almost identical and are therefore clonal in nature. This may best be explained by principles of population genetics, especially the concepts of genetic drift and bottlenecks. In population genetics, genetic drift is the idea of certain alleles becoming fixed within the population due to random chance, or a so-called sampling error. In mitochondrial terms, mitochondria undergo constant turnover via lysosomal degradation, fusion, and fission, especially in response to metabolic needs. Thus, through random chance, certain mitochondrial mutations may be lost from a population of mitochondria. This creates a founder effect, in that only the currently present mitochondria will be able to repopulate the mitochondrial population. If those mitochondria carry mutations, all subsequently generated mitochondria will be clones of that original founder mitochondrium and thus will carry the same mutation. One interesting mechanism that may further speed up mutation accumulation via drift is that mutated mitochondria experience a drop in electron transport chain efficiency, which in turn stimulates mitochondrial replication in order to increase ATP output. Of course, since decreased mitochondrial efficiency is due to mitochondrial DNA mutations, mitochondrial replication only leads to further expansion of the mutated mitochondria, further exacerbating the problem and once again possibly leading to fixation of the mutation in the entire mitochondrial population. This random genetic drift in clonal expansion is expected to have consequences for tissue health and aging. Genetic drift by its very nature is a slow process, slowed even further when applied to highly dynamic populations, such as mitochondria. Detrimental mutations may take years to fix within a cell, and therefore tissues become mosaics of cells either carrying a healthy or a damaged mitochondrial population. Experimentally, this is often visualized by cytochrome C oxidase staining, as we have discussed earlier in this module. Cytochrome C oxidase deficiency has been best described in muscle cells, neurons, and the crypts of intestinal tracts. In muscle fibers, CO-deficient zones tend to appear in clusters, quite possibly clonally derived clusters. While they only constitute a very small quantity of muscle in young individuals, this number rises dramatically with age, but even then remains usually within the single digits. However, due to the clustering nature of these CO-deficient zones, muscle fibers are more prone to breakage at these zones, which leads to the degeneration of the entire muscle, which in turn may lead to age-dependent sarcopenia. In neurons, the number of cells with DNA mutations increases dramatically with age as well, but to a much higher level than in muscle cells, reaching up to 80% of the cell populations by 80 years of age. The most affected neuronal subtissue is the substantia nigra, a part of the basal ganglia which is important for motor control. Incidentally, this is also an area of the brain affected by Parkinson's disease. Thus, symptoms overlap: muscle rigidity and tremors, and general difficulties in coordinated movements. It is thought that these substantia nigra neurons, due to their extraordinarily high basal metabolic activity, are particularly susceptible to mitochondrial mutations. Even in highly mitotic tissues, such as the cells lining the intestines, mitochondrial mutations may accumulate over time. The origin of these mutations therefore lie within the crypt stem cells, which are needed to constantly renew the tissue. Once an intestinal stem cell has acquired a mitochondrial DNA mutation, this mutation will clonally expand due to stem cell regeneration, and eventually a significant number of cells with damaged mitochondria will be present in the population, approximately 15% by the age of 80. This could partially explain the increased incidence in colon cancers in the elderly. Interestingly, when mitochondrial biogenesis in the colon is experimentally induced, fruit flies have been shown to be longer lived. Are mitochondrial DNA mutations therefore drivers of aging? One way to address this issue is to create so-called mitochondrial DNA mutator models, for example, by deleting the mitochondrial DNA polymerase. These animals, shown in panel A as the red line, have markedly decreased longevity and increased mutational load. They also show many signs of premature aging, for example, kyphosis, which is the hunchback phenotype, and to a lesser degree, graying. Interestingly, mice in which pieces of mitochondrial DNA have been deleted do not quite recapitulate these very same phenotypes, suggesting that the relationship between mitochondrial DNA damage and aging, while tantalizing, is really not quite as clear-cut as hoped. Check your knowledge. Explain the cause of mitochondrial DNA mutations. Describe the population genetics of mitochondrial DNA mutations. Describe the consequences for tissues on organismal aging of mitochondrial DNA mutations.